So first, a couple of slides presenting the uniqueness of and challenges we have over there in the Southern Great Plains, in North Texas and Southern Oklahoma. The basic three grazing systems over there is the warm season or C4 grasses that are productive in summer from more or less May through September. And then in the cool season period, the major, of course, is dual-use wheat or grazing wheat, planted in fall. If there is enough rain, there may be some grazing over the winter, and then mostly early, late winter, early spring up to March, when the cattle is taken off from the dual-use wheat. In the past, up to about end of 90s, there were introduced traditional cool season perennial grasses that also fit into the winter grazing system, but after 2000, more or less, with the changing climate, they disappeared. They cannot be grown over there. So we used this opportunity and tried around early 2000 to introduce summer dormant cool season grasses. And you can see they produce very good forage in fall, when it's raining, of course, and then through winter and especially in spring until early summer. I also showed here the bimodal precipitation, the historical bimodal precipitation pattern in our area. This pattern does not really exist anymore. In the recent years, it was shifting from May to June and September to October, the peaks, and recently it's just random. Changing temperatures and precipitation for the Vernon area. The observation started in 1935, so I tried to plot here differences or from the departures from the normal in temperature, which will be the red columns, and precipitation, which will be the blue columns over the years. And you can see very nice cycling pattern with the warming climate during the 40s and 50s. This was the Dust Bowl area in the United States, especially in the southern plains. Then cooling pattern of the climate in the 70s and 80s. And then again, warming climate pattern right now starting in more or less early, late 90s, early 2000s. Okay, so the major characteristics of the changing climate is increasing temperature, decreasing precipitation, abrupt rainfall pattern, and increasing frequency and severity of drought. Like I mentioned there, the wheat is the main source of winter forages. This is for the cool season grazing period. The traditional summer active cool season perennial grasses are not existing there anymore. And now the summer dormant cool season grasses introduced around 2000. They are very well adapted and persistent in this environment and have excellent autumn and spring forage productivity. They evolve in the Mediterranean climate. I will show later the map not only of Northern Europe, also in California. They express endogenous mechanisms of summer dormancy regardless of soil moisture. They have active autumn-spring growth and complete or incomplete dormancy in summer, depending on the species. Induction and release of summer dormancy is most likely controlled by temperature and day length, although nobody stated this to be totally sure about that, but we did suppose that both of these environmental factors have a very high effect on this. Often they can form surviving organs, not all of the species, but some of them do, in form of corms or bulbous structures at the basis of the tillers. Very nice characteristic of them is, especially for Fescue's orchard grass, the seed will not germinate if the day length is about 12 hours. And this is the distribution and origin of summer dormant cool season grasses with the red squares or rectangle. I showed origin in the Mediterranean basin of Europe and North Africa, but there are also native summer dormant grasses for California and for Texas, believe it or not. Texas does not have a Mediterranean climate, but there are two species over there, Nazella and Poa, that are summer dormant. And current research is, occurs in several countries in Europe, Southern Europe, in United States, in Texas and Oklahoma, in Argentina, and then Australia and New Zealand. For example, a tall fescue distribution, the two groups, the northern line depicts the summer active types, the red one, the summer dormant types, just for orientation where they occur. In United States, I don't want to go very briefly through this, but we started in cooperation with, now it's Grassland's innovation, but it was Ag Research at that time. In 2000, the program started in Vernon in Texas, but also at the Noble Foundation in Oklahoma, in northern Oklahoma. Major highlight for my program was in 2004, we got in cooperative research with the Hebrew University of Jerusalem on this type of grasses. And then in this year, we'll be releasing the first summer dormant tall fescue, Jonathan. Now about my program. So we work on three species, developing cultivars of three summer dormant grasses, tall fescue orchard grass and the heat-tolerant perennial ryegrass. In fact, the perennial ryegrass is not really summer dormant, but it behaves like. And now introducing the new cultivar Jonathan. Jonathan comes from Golan Heights in Israel, and I presented a few pictures. It grows in rocky soils in a hills area, and as you can see also they can get some snow over there and colder temperatures in winter. The elevation and annual precipitation are very close to the area we'll work with this grass in Texas. So this is very similar. And just going briefly through the breeding history, the program was, has been supported from the beginning by Grassland's Innovation and PGG Wrightson. And this new cultivar will be commercialized in the United States by Turner Seed Company from Breckenridge in Texas. The original accession was infected with the epichlor, or formerly known Typhodium endophyte. It was a toxic endophyte, so it was eliminated right at the beginning, and all following recurring selections were done on endophyte-free material. We selected the plants briefly for wide leaves, increasing tiller number, strong tillers, and resistance to leaf rust. And the resistance to leaf rust was done in Texas, and then again on the last cycle in New Zealand for the races they have over there. This and seed certification, application for seed certification was filed in September. By now it's already approved. Few pictures about this. On your left side you see the tillers of Jonathan. What is very unique for this particular selection, that it forms, they are not morphologically bulbs, but bulbous structures at the basis of the tillers in spring. And we believe that this is one of the mechanisms that allows for this particular grass to survive summer drought, and very well, much better than other summer dormant tall fescue. On the right side you see the same tillers of Fletcher, which originates from a different area than Jonathan. Jonathan is also very early, much earlier than other summer dormant grasses. You see here, this is a blooming, so Jonathan will be the red line. The other three checks, Fletcher, Chisholm, and Jessup, summer dormant, are a bit later, and the summer active check is totally late. Other morphological characteristics differentiating Jonathan from other summer dormant tall fescues, as I mentioned, it has wider leaves and longer leaves. This will be the red numbers. And The flag leaf is also wider than in other summer dormant grasses. And there are also differences when you look at least between Fletcher and Prosper and Jonathan in seed weights and some other morphological traits. This is an example of a third season annual year, total year for the season, for the growing season. On 2 locations we tested, in Vernon and Breckenridge. So you can see that with time, the other summer dormant grasses like Fletcher and Chisholm and Prosper will, the yielding will decrease because of the response to the constant drought over there. But the Jonathan will be maintaining the productivity, and eventually with time will be better than any other cultivar. Jonathan is also much more dormant than other, at least the 2 summer dormant checks that we have, Fletcher and Prosper. As I mentioned, especially in tall fescue, there is no, the dormancy is not complete. There will be always species or accessions or cultivars or accessions that will vary in the dormancy expression. In contrast, for example, to orchard grass. Orchard grass is very high dormancy score, so even occasional rains in summer or so will not break the dormancy. In tall fescue, if it starts to rain too much, at least in our area in summer, the dormancy may be broken very, very easy in some of them. There has been always a lot of questions about what's going to happen if those grasses get out of the plots to the rangeland over there, what's happened there in Texas or in southern Great Plains. So we right at the beginning, we did some research. This is greenhouse research, analyzing the aggressivity of summer dormant versus summer active and alfalfa. And we found out, like you can see on this graph, that regardless of pretty much of soil moisture, summer dormant grasses are not able to compete with alfalfa in summer over there. So we think that our rangeland in majority are summer active grass, C4 grasses that grow actively in summer. So even if this escaped, it has no chance to compete with them. Again, but there may be other aspects of the invasiveness. As I mentioned, there are 2 summer dormant cool season grasses in Texas, native ones. Texas wintergrass and Texas bluegrass, as well as in California, we have 3 of them. So if this is going to grow in California, for example, we don't know what the ecological effect may be if they escaped from the pastures. So in conclusions, traditional summer active or continental types of cool season perennial grasses are not adapted anymore to our environments because of the changing climatic patterns. Summer dormant or Mediterranean cool season perennial grasses have been proven persistent and adapted to climatic conditions of the southern Great Plains, and this is backed by long-term research done at our station as well as the Nobel Foundation. We have about 20 years of data right now to prove that they are adapted to our environment. Jonathan is one of several improved summer dormant tall fescue cultivars that we currently develop with, at our station in collaboration with Grassland's Innovation and PGG Wrightson. Thank you. Questions?
I'm sorry that we don't have time for questions, but perhaps you'll be around, Darius, for. So catch him individually if you would, and we need to move on to the next speaker. Thank you very much, though. While we're getting set up, I want to announce again that if you had intended to catch the last session in Grassland Sustainability yesterday, that person flight got delayed, but they are here today, so we're going to add it on to the end. So if you wanted it yesterday and missed it, they'll be here at the end today. Our next speaker is John Caridas on genetic modification application. I practiced saying that for forage and grassland production systems. Turn it over to you.
Thanks very much. Look, this may be a fairly controversial topic for some, but I also believe it may provide some of the solutions that we're going to need if we're looking at solving some of the issues that were raised on Sunday night by Peter Ballastat regarding the importance of grasslands for feeding an increasing global population. Firstly, a declaration. I'm not a molecular biologist. I am a humble, or I was, a humble plant breeder. I now look after a company which invests in, as and involved in, a number of projects involving genetic modification in forages, and I'll briefly cover off on those as we go through the talk. So this is what I'm going to talk, this is where I put genetic modification. It's obviously transgenesis, gene stacking, new breeding technologies, which includes gene editing, and RNA gene silencing. Over the past 25 years, GM technologies have been used to provide both human food and animal feed. The adoption rate, as shown in this graph, has been outstanding and probably faster than any other recent crop development or technology. Just in 2022, there were 824 approvals allowing the cultivation of GM crops for 8 types of GM traits across 26 countries. So it's very, very widely used around the world. The table on the right here actually goes right off the bottom of the slide. But the most common traits that have been used in the past in crops have been glyphosate tolerance to control weeds and genes expressing insecticidal proteins. There's been an increasing number of other traits being used that include tolerance to abiotic stresses, disease resistance, modified product quality, and pollination control. However, there have been very, very few examples of GM technologies being used in forages and grassland species, certainly commercially produced. There are 2. Roundup Ready alfalfa and Harv Extra also in alfalfa. Yet surprisingly, 70 to 90% of GM crop production is currently used to feed animals, not humans, to feed animals. And the largest users of these GM crops for animal feed are the United States, where we are here, China, and surprisingly, Europe, considering their regulatory ban on growing transgenic plants in Europe. So just stepping back and saying, so GM technologies is a means to an end. So what are some of the challenges confronting grassland agricultural systems at the moment where we might use GM technologies to solve those issues? There's economic, environment and society issues that are impacting managed grassland systems, and we've heard about some of those in the plenaries and keynote speakers in the last 2 days. Additionally, climatic events and changes, competition for land use, so we're not inventing any more land. In fact, the amount of land, as we've seen in some of the previous talks, that can be used for agriculture is in fact decreasing. So we've got to be more efficient in terms of the food and crops that we are growing. There's biosecurity incursions. Energy use efficiency is still a challenge. Changing consumer preferences. Where does the synthetic meats, plant proteins fit into this whole scheme of things? And the creation of new value-added food products. Well, genetically modified traits can potentially benefit grasslands systems, but I'll just make the point that GM technologies aren't going to solve all apparent challenges that we have in front of us for grassland systems, but they may potentially alleviate some of those. And here I've listed some of the potential plant traits that genetic modification may provide a benefit for in the future. And they're listed here largely because they have been shown to be the case often in laboratories or contained experiments in glass houses. But certainly some of these solutions around disease resistance, insect pest resistance, drought tolerance, improved winter hardiness, salinity tolerance, etc., could potentially be solved through the use of GM technologies. We mustn't also forget that our plants live with a variety of beneficial microbes, and so it is potential that we could have, in fact, improved some of those in the way they interface and provide benefit to foragers as well. We're involved currently in 3 genetic modification forage programs that I've listed here. I'm not going to go into any of these in detail, but we have managed to get condensed tannin expression in white clover. We've also managed to do it in alfalfa as well. But in terms of white clover, this has the potential to reduce methane production by probably about 15%. It will reduce urinary nitrogen, bloat, will be something of the past. It reduces internal parasite burden and improves animal productivity. I mentioned about using GM technologies and microbes. Well, Epicloe endophyte, we heard a little bit about that in the previous talk in some of the tall fescues. They are incorporated into plants because of the secondary metabolite pathways they produce in planta. But we only often know about the end secondary metabolites that's produced in these pathways. We know very little about what the intermediate compounds that are produced in those pathways actually do. So we can use gene editing to manipulate those pathways to get increased expression of those intermediary compounds to determine whether in fact they have some potential benefit to the plant and to the animal that's eating them. And lastly, the high metabolizable energy ryegrass. This is where we increase the lipid content in the ryegrass and protect that lipid with a coating. And it's been shown that this in vitro experiments can reduce methane production by 10 to 15%. Now, ideally in the future, what I could envisage is we'd have our condensed tannin white clover and our high energy metabolite ryegrass grown together as a transgenic sward, which would reduce methane production significantly. These are data from in vitro experiments that have been done looking at methane production and the reduction in methane production due in the first instance to condensed tannins in white clover over a 6-hour period, where you can see that the high CT reduced methane by 17%, and that's compared with Lotus caniculatus, which naturally produces condensed tannins, reduced methane by 16%, so very comparable. On the graph on the right, over a period of 24 hours, shows that the high metabolizable energy ryegrasses, which are the black and green lines, which are the red and yellow lines, reduced methane production considerably compared to the controls, which were the black and green lines. However, it is a controversial topic and there are consumer concerns, rightly or wrongly. And some of these concerns include food safety linked to potential toxicity and allergenicity, environmental risks associated with the potential of gene flow from that transgenic plant, adverse effects on non-target organisms, and the evolution of resistance of weeds and insects, and genetic perturbations leading to the production of unintended compounds, new diseases, and antibiotic resistance. But most of these can be tested, and most of them can be either determined to be a problem or not. There are some other concerns around labelling and consumer choice, intellectual property rights, ethics, and coexistence with non-transgenic crops. Regulatory challenges. Many GM-regulated traits and forages have been developed by public institutions and are not linked to end manufacturers and suppliers of that seed. Those institutions are really incapable of taking them through national regulatory processes because of the expense. Potential risks associated with GM crops should focus on. So I'm not saying we shouldn't have regulation, we should. And they should focus on human and animal health and welfare impacts, including testing for allergenicity. They should be looking at impacts on beneficial non-target organisms, and they should be aware of the gene flows from GM crops to other weedy species. The problem with a lot of our forage crops is that they're outcrossing, and there is a lot of weedy populations that those genes can move to. So coexistence of GM and non-GM cultivars of outcrossing forage species is an acknowledged challenge. It's not insurmountable, but it is acknowledged. So in conclusion, while I haven't covered off on it explicitly because of the time, there are inconsistent approaches to regulating GM plants across different jurisdictions. Many countries allow the consumption of GM food and feeds, but do not allow GM plants to be grown, which simply creates confusion. And I've given 2 examples there of Europe and New Zealand. GM technologies have been extensively used in providing many benefits in crop plants, which are most often used for animal feed. There still needs to be a balance of risks against benefits to ensure adequate testing is undertaken before we do use these GM feeds commercially. And this should include monitoring animal health effects on non-target organisms and understanding the impact of gene flow. Thank you.
Thank you. We do have time for a few questions from the audience.
At what point do you think the scientific community is going to come up with a different nomenclature for genetically modified organisms? The minute people see genetically modified and don't understand it, they envision a Frankenstein model.
Yep. Yes, it's very unfortunate, the publicity that generated in the late 90s and early 2000s, and I really do not know how we convince the public that we're not talking about Frankenfoods here. We are talking about well-constructed and understood gene changes that we can actually measure and determine. You know, it's interesting, if we look at regulation, we can mutagenize plants and microbes using chemistry and chemicals and radiation, and they can be put out for anyone to use without any regulation at all. We have no idea what those changes are to the genes or what they're doing, but we can put them out in the environment and they're not regulated. Yet if we gene edit a plant and we know what that gene does and we know the result of that gene edit, we still have to go through, in many jurisdictions, including New Zealand and Europe, a regulatory process to determine that it's safe for use. So it's those inconsistencies that we need, firstly, to address from a regulatory point of view, and then we need clear communication to the public that we have this relatively under control, or as best that we can. There's one right down the back if you've got time.
Yeah, question about freedom to operate on, IP issues. Now that the technology has been around for a couple decades, most of those are utility patents, so they're expiring. I guess what's your opinion on, as these utility patents expired, will that incentivize smaller players to get into the market?
Yeah, it's a really good question. And the company I look after is a really small player. And I've got a meeting next week, or it's the week after, with Corteva to try and negotiate a commercial license for them for gene editing epicloiendophytes. And I tell you what, I'd rather bang my head against a brick wall, but somehow or other we've got to do it. So you might be right that some of those old utility patents have gone, but a lot of these big organisations have got other patents which catch us still today. And gene editing, which has really come into its own in the last decade, is really the way of the future, I think, rather than transgenesis. And those patents are still alive and well. So it's not a done deal that we can just have freedom to operate. Thanks.
Thank you. Real quick again, for those of you that didn't hear the announcement earlier that joined us for this session, if you had intended to catch the last presentation in this track yesterday on alfalfa, that person finally made their connecting flight and will present at the end today. I'd like to go ahead now and introduce our next speaker, David Hume, Stability and Purity of Endophyte Infection in New Zealand Ryegrass Pastures.
Thank you, Keenan. Now, it'll be no surprise to most of you that perennial ryegrass is the dominant sown grass in New Zealand. Well, what you may not know is that it's typically infected with a fungal endophyte. And you heard Darius refer to it briefly and John Carradis. And the key thing about this fungal endophyte is that it imparts to the grass plant increased resistance to biotic and abiotic stresses. That's really important in our environment. So today, most of the elite proprietary perennial ryegrasses are sold with a selected endophyte. And the key thing is that many decades ago, these ryegrasses had the standard wild-type mammalian toxic endophyte. And these selected endophytes have really displaced that standard endophyte. So for example, in a dairy sector, we know that the standard endophyte can result in up to 9% milk losses production, animal disorders such as ryegrass staggers, and exacerbating heat stress. So these selected endophytes, they reduce or eliminate that animal toxicity, and to varying degrees, they impart a positive agronomic advantage that we want. So there's not a trade-off going on here. We can get the best of both worlds. Now, what actually is an epicloe fungal endophyte? So here, if we take a scanning electron microscope view of the inside of a grass leaf, so the cell contents have been removed, but we can see the grass cell walls here. And attached to those walls are the strands of the hyphae from the fungal endophyte. Now, a key thing is that the fungus is not trying to enter those cell walls, so it's not going to become a disease. And equally, the grass plant is seeing that it's a friend. It's not setting up some adverse reaction. And these grass epicloe fungal endophyte associations are classified as mutualistic. So there's benefits for both the grass and the fungus. They're obligate. The fungus can only grow or live inside a live grass plant or seed. And it's asymptomatic. So we need laboratory tests to determine whether it's present or not. Now, in this particular study, we know there are significant benefits for farmers to use selected endophytes in ryegrass. Now, we've intensely monitored these associations within experimental settings, but not in the commercial scale. We do know that pastures could become contaminated with the standard endophyte again. And we've done some experimental work, you can see from the graph here, to show that standard endophyte can outcompete some of our selected endophytes. So there's been concerns have been raised around whether the seed supply chain and the actual pasture renewal process on farm have been delivering these selected endophytes in a commercial setting. And then the question is the standard endophyte just going to come in and outcompete and undermine the benefits of our selected endophytes? So in our study, we looked at pastures in the North Island where 5 ryegrass cultivars had been sown with one of 3 selected endophytes. And then into the eastern South Island, 2 ryegrass cultivars and just the 1 sown selected endophyte was in these pastures. So we went in annually and sampled ryegrass tillers. We determined the frequency of endophyte infection using immunoblot. And then using a subset of those tillers, we determined what endophyte strain was present using a DNA test. So, right into the results. You can see both the average for the North and the average for the South Island is quite high. And there is a very small increase over those 7 years, statistically significant, but actually quite small and not particularly important because the levels are so high. Now, I'll give you a few more details because there's some exceptions to those averages. So in the South Island, we seem to have a group of pastures which were close to that 90% infection, but also a group that averaged in the 86%. And 2 of those pastures were below that 70% desired minimum that we have. And one of those we know was on an organic dairy farm. They were not able to use synthetic herbicides to kill the previous pasture. And that actually happened to be my sister's dairy farm. I found out many years ago, later, sorry. So when we look at this issue of whether there are non-sown endophytes or contaminating endophytes, you can see on average for both the North and South Island, they were quite low. In the North Island, they actually increased over those years. Now the key thing is which strain of endophyte it was. And as we had predicted, standard endophyte was the major contaminant. And you can see that it did increase over those periods. Not particularly much, but we need to look at some of the individual farms. So in the North Island, we have most of the pastures, 5% contamination, and no change over those 6 or 7 years. But you can see we had 3 pastures, which went from 19 to over 50%. And at least for one of those pastures, we know that uncertified seed was undersown there, and that could have introduced some more of the standard endophyte. And in the South Island, my sister's organic farm stood out again. In this case, it wasn't actually standard endophyte. It would have been leftover AR1 endophyte from the previous pasture, was 25%. Now we had one additional North Island pasture which was sampled for 7 years. Now in this case was inadvertently sown in a seed line with low viable endophyte. So it started at 24%, and this one increased 4% per annum to be 52% at the end of that 11 years. And you can see again it was due to the standard endophyte increasing. In this case too, this particular product has 2 endophytes, NEA2 and NEA6, and the ratio of those 2 endophytes didn't change over time. So in conclusion, and I'll summarise what I've just told you. So in general, the industry has been able to successfully deliver these selected endophytes through the seed supply chain, through the pasture renewal process, onto dairy farms. And those selected endophytes have stayed at high proportions, on average, in most of those farms. Now there were a minority of pastures which didn't reach our 70% desirable level. And as we may have predicted, that standard endophyte was the major contaminating endophyte, and in some of those pastures, not all, but some of them increased over time. No real concern in the South Island because contamination levels were so low. Except remember those 3 North Island pastures increased over time and were over half of the tillers with the standard endophyte. And at that level, we would expect them to be standard endophyte impacting on the animal productivity and also the animal health. So they're not a good situation to be in. Now remember, this is just an on-farm study. We couldn't necessarily understand all the drivers, but at least for 2 of those farms, we could perhaps attribute the outcomes, or the poor outcomes, to 2 of the management practices. Now, if you want to know more about epicloe endophytes, not only in ryegrass, but in tall fescue, I suggest that you come along to the session on Thursday afternoon, a thematic session around a model to reduce and eliminate endophyte toxicities in temperate pastures. So thank you, Peter.
Thank you. We do have time for a few questions if the audience has any.
So I saw changes in the endophyte levels on some of those pastures. Was the endophyte stable in all those pastures throughout, or did it decrease?
Oh, it's very stable, actually. All our lines literally just kept on going up gradually. There was obviously some variation, which is often driven by the number of ryegrass tillers that we can take. But they were really quite stable, and that was quite a pleasing result to have. Well, I think Randy's got another one.
So what was what is the source of the wild type increase again from those pastures where the wild type came from or increased?
Well, so we don't necessarily know what it was, but we know that the dung from dairy cows can carry seed of ryegrass in that dung. It germinates in that dung and that manure, and those seedlings have endophyte in them. So that's one particular source that can happen. And so that's not a bad management practice, but we do know, like for the organic farm without the herbicide use and undersowing with uncertified seed, that were two practices which probably contributed to the contamination. And I think there's the issue is that there's multiple paths happening out there for contamination to occur, but we don't really know what they are in any particular farm or any particular season. And so this monitoring has at least given us the end result and some confidence that we're not rapidly losing these selected endophytes in terms of their percentage in the pastures over time.
We've still got another minute or two left if we have further questions. Or perhaps for our first speaker, Mr. Malinowski.
Yep.
Dr. Hume, what would were the perceptions of producers when these new endophytes were first introduced, and how long did it take them to kind of make that transition?
Good question. And actually, John Karras, you might like to be better positioned, John, to...
So the first release in New Zealand was in 2001. It was AR1. And by 2007, it made up 70% of the proprietary ryegrass sales. So the uptake was absolutely incredible. Yep.
I think it was also a combination. There was farmers and extension people and seed industry people, they were all on board. There was lots of information, lots of education over years and years. And they just saw these advantages. New Zealand farmers, if they think there's an advantage there, they will uptake endophyte technologies really rapidly. And it was available across a wide set of cultivars. So as an organisation, John commercialises our particular products, and it was available across a lot of products all at once. So just all the right things happening at the right time to get the technology uptake.
Thank you very much.
Yeah.
As folks have cycled in and out, I do want to announce that if you had anticipated the last presentation in this track yesterday, that speaker wasn't here yesterday. She finally made it in today, or in time for today, so she will present at the end of this session. Our next speaker is Jesse Morrison, Strategies for Seed Quality Improvement in Eastern Gamagrass, Tripsacum species. Close?
That's good enough.
Okay.
Thank y'all for being here. So unlike everybody who's talked before and my graduate student who's going to talk next, I know next to nothing about endophytes. Sadly enough for my graduate student, she did an endophyte project. So you'll get more endophyte talk after I'm done. For the last 10 years, I've been working with Eastern Gamagrass. Eastern Gamagrass is a North American native warm season perennial grass. Grows into big, large, donut-shaped clumps as it gets older. The middle of the clump sort of dies out. It's a close relative of maize, which doesn't really matter for the purview of this conversation, but it's also monoecious. So if you see the picture of the Seed head up there, those are the actual seed on the plant. That's where the female flowers were. So you've got male flowers above those. They have since senesced and fallen off. And so as these female flowers seed ripen, they'll just dehisce and fall off the plant. You can also tell from there in that single inflorescence you might have 20 seed, and that's pretty common, anywhere from like 7 to 20 seed per inflorescence. So not a prolific seed producer in the, the world of grasses, even native grasses. In the wild, though, you'll find gamma grass in diploid and polyploid, mainly tetraploid cytotypes. That's important because diploid cytotypes, by and large, are sexually reproductive. They're great for breeding programs. Tetraploids, by and large, are apomictic. So if you're not familiar with apomixis, it might be the worst thing ever for a plant breeder. It's essentially like plant cloning via seed, right? It's hard to make improvements. It's hard to select for specific phenotypic products. It doesn't eliminate the ability to improve varieties, but it makes it really difficult. So for this species, for what we're talking about today, we tried to just focus on diploid cytotypes. So eastern gamma grass is really widespread from the Great Plains states in the Midwest all the way to the Northeast and the Deep South. These pictures are from a beef grazing operation in the Finger Lakes region in New York. When it's the warm time of the year, they graze only eastern gamma grass. They graze cool season mixed grasses and legumes in the winter, and they feed hay, but in the summertime, it's gamma grass or nothing. The popular cultivars of gamma grass you'll see on the market today, most come from the Great Plains states, Kansas, Nebraska, Oklahoma. So gamma grass has lots of uses. It is very versatile, whether it's for conservation and filter strips or for phytoremediation. There are some neat ornamental varieties that come out of Florida. It's important for inclusion in wildlife mixes, for habitat, for food, but it's also a really high quality forage. Janet Grabowski had a study that they published in 2003 where they were comparing eastern gamma grass to Bermuda grass in irrigated, non-irrigated situations. You see that they got good crude protein, good ADF and NDF depending on management. Yield is really good, especially when it's irrigated. Their study, their 2-cut gamma grass out-yielded and had higher quality than a 3-cut Bermuda grass system under the same management. Pat Kaiser in 2020 put out a study where they compared eastern gamma grass and sorghum Sudan grazing replacement heifers. Had better gains, had cheaper gains per pound, and better gains per unit area using eastern gamma grass. So it sounds great, right? Why are we talking about it? Because the one place it can't really compete is the establishment world. So establishing eastern gamma grass is remarkably difficult, mostly because of the seed structure itself. So what you've got is, you can see from this scan, you've got a hard, woody, cupulate fruit case that the embryo lives inside. So it's a combination of the rachis internode and the glume fused together in this hard, woody case. So it's great for cleaning seed. It can take a lot of abuse. You can rough it up. It's not really going to damage it at all. But when you go to plant it, it's very difficult to get a live plant out of that seed. This lower picture is something I see in the lab a lot. You get radical emergence. You never get a coleoptile out. You can cut those seed open and look at them and see that the embryo woke up, endosperm started getting dissolved, it started trying to elongate, but this little part of the coleoptile right here can't make it out of that crease. So the solution to that is commonly cold, wet stratification, right? We soak seed in water for 24 hours, we take them out, let them drain, we put them in a cooler for 6 weeks. That 6 weeks of wet and cold kind of breaks down that fruit case. It helps leach out some of the endogenous chemicals that are going to cause some chemical dormancy issues, and it generally increases germination. But then you pull that seed out of the cooler, you put it on the UPS truck, you ship it to the buyer, they're paying for shipping water, and when they get the seed on the farm, they got to plant it, immediately before they have fungal issues or whatever else. So it's not an ideal situation for any producer looking to establish gamma grass. The other big issue, this is a seed tag off of a bag of seed that I bought for some of the research we were doing, and I have tried my best to protect the guilty party involved. But anybody who ever deals with seed, look at that seed tag and tell me what's missing. There is no % germination. Now, if you were a producer and you bought a bag of gamma grass seed and it showed up at your door and they didn't even put a % germ on the tag, wouldn't you be concerned? Right? Like, yes. Yeah, I get it. I get it. They're 93% alive. That's great. But how many of them are going to germinate? If they can't tell you that, that's a problem. So, my objectives for the last almost decade have been to screen diploid germplasm for rapid germination. Just find some plants that'll make some seed that'll germinate really quickly without any treatment. No chemicals, no hormones, no peroxide, no soaking for 6 weeks, no nothing. And see if we can build a population of those plants. So when I started this, like any enterprising young professional in the native plant world, I thought the best thing to do would be drive around the southeast and collect wild plants. So that's what I did. I drove around 9 southeastern states. I collected 500 some plants. I dug them up out of people's yards, off their farms, people who had eastern gamagrass and didn't know what it was, people who could assure me that they didn't plant it in the last decade. I went and collected everything I could find. Brought it all back to Starkville, Mississippi. I planted it in the nursery. And the first thing I needed to do was make sure I had diploids or tetraploids, and I could throw all the tetraploids out, and I could keep all the diploids, right? So we do some flow cytometry work. All we're doing in flow cytometry is looking at how much DNA we have in our in the nucleus, right? So we have a standard diploid, we have a standard tetraploid. We used P's just for another standard to make sure that all our measurements were right. So we ran this machine. We had a standard here and a standard here. We would run an unknown and just look for a new peak. And if it came up here, it's a diploid. And if it came up here, it's a tetraploid, right? So we ran 547 samples through the flow cytometry machine. And everyone was a tetraploid. Everyone. Did I say everyone? Every single one. So, my grand plan, that failed. That strategy did not work. I said strategies in my title. This one did not work. I have a great nursery of tetraploid gamma grass if anybody's interested, though. So, we dropped back, we punted, and we just went to the internet. We fired up the Google machine. We looked for whoever was selling gamma grass seed, right? We have named varieties of diploid gamma grass, so we bought every diploid we could find on the market. That bag of seed with no germ on the label. That's one of the ones I bought. So we just started screening seed. We could fit about 3,000 seed on a big metal tray. We would just wet paper towels, put them in a germ chamber, and see what grew. If we got anything that germinated in 7 days, we kept it. Everything else we threw away. So it took us about 90,000 seed, and we got 100 seedlings that germinated in less than 7 days. And so those became our original nursery population. Gamma grass is also notorious for not flowering in its first year. It has to go through one good vernalization. So we were germinating these in October, November, December. We were getting them into the greenhouse around January, and we kept them in warm temperatures, long day lengths. We kept pouring fertilizer to them. We bulked them up as best we could. We'd get into February and March. We'd start introducing them to ambient day length and then ambient temperature. We'd let them get cold. We'd let them go ahead and senesce, and then we'd plant them in the spring. So we plant those senesced plants in a crossing block, gave them a little bit of fertilizer to start the year, and then in June and July, you can see this tiny little clump. This is a 1-year-old plant, but it's making seed. We started harvesting seed. We let everything open cross, and because seed were so precious and we got so few, we started tying these bags on every seed head we had, right? So the guys at the farm all had questions like, what on earth are you doing in that block back there? But it helped us keep seed and keep identities straight, and so we kept our mother plants ID'd. But this is our first germination. So we started, like I said, we made a base population out of peat and iuca. I include some monotypes in here, but it's not like they did anything anyway. So our base population, cycle 0, we stratified for 8 weeks. So we took a set of peat, a set of iuca, 6 reps of 100, stratified them, germinated them just to see what we'd get. And still, after stratifying, pretty poor, right? This is 14 days, and our highest daily germination rate was 2.5, 2.6%. Pitiful. But we took that same seed lot, didn't stratify it, and we got that. Right, so this is why it took us 90,000 seed to make a population of 100 plants. So our first year, we've got cycle 0 in the blue, cycle 1 in the black, and cycle 2 in the orange, or the gold. Over here is mean daily germination, that's for the bars. And over here is cumulative germination across 14 days, that's for the lines. So you can see we had a significant difference between cycle 1 and cycle 0, and a significant difference between cycle 2 and cycle 1. I didn't put the bars on there, I'm sorry. You can trust me, right? They were significantly different in our first year. So these two plots by now are 2 years old. This one's 1 year old. So we get to when it's 2 years old, you can see our scale's 12%, still hadn't changed. These two don't really move much from year 1 to year 2, but we get a big jump in cycle 2. And these aren't planted too far away from each other, right? So we weren't real concerned about environment being a thing. But we realized real quick that 100 plants in a nursery is not enough to make enough seed to keep screening for another cycle, right? So while we're doing this, we're continually screening more seed to add plants to cycle 1 and continually screening more to add to cycle 2. So it takes us 2 years, 3 years, and then we finally make it up to cycle 3. And if you notice now our scale's changed. Now we're up to 40% cumulative germination in 14 days. And these red bars are cycle 3, and we're starting to see an impressive shift to germination earlier than 7 days. These same seed lots now, if we stratify those and germinate them, we're looking at 70 to 80% germination. So we're not as far as we would like to be. And these are several years old. We have more germination data, I just haven't had time to look at it. So my apologies for the data being a little bit old, but all in all, I think it's a success story for Eastern Gamagrass. Fairly straightforward results. We started using pollinators. We started buying bee boxes, and that seemed to help seed quality and seed set a little bit. And trying to identify structural components of that hard fruit case. Like I said, you've got the rachis internode and the gloom. The gloom's kind of like the top on the canoe. Trying to select for plants with smaller glooms, shorter glooms, is something we're into now, but we've started that, the last year, so I don't really have any results from that yet. But that's all I got. Thank you.
We do have time for 1 or 2 questions.
Hello, I imagine you evaluated the apomictic ones, the tetraploid also? Just to see if you find something miraculous?
Yeah, and we do have some tetraploid varieties that have decent germination. But I honestly haven't moved forward with any of those in any kind of meaningful way. We trialed them. We mostly trialed them for forage quality and environmental tolerance, so like cold tolerance, heat tolerance, that sort of thing.
Yeah, because you could duplicate the sexual ones and cross with the apomictics?
Maybe. We've looked at the apomictic gamagrasses as just if we can develop a good enough population to make a synthetic blend from, we could do that. But I haven't focused on them as a pollen donor at all for anything.
Thank you. I hate to cut off questions when we're out of time, and I'm told that I have to be really strict about that, so I hate it when we don't get to have further discussion. I would like to introduce our next speaker. I'd also like to thank her because she's been handling the computer techno stuff, and without her, you all would just be listening and not seeing anything. So Brenda Hale, thank you. Her talk is on isolation and culture of whatever that is for reinfection of endophyte-free southeastern wild rye. Thank you.
Thank you for the introduction. I just want to thank everybody for being here, and I know we're going to have a blast as I go through my master's degree research. So as he said, this is isolation and culture of epicloe for reinfection of endophyte-free southeastern wild rye. So just to introduce this project, here in the United States, we rely heavily on our livestock as an important food source, and so the perennial-based pastures that these livestock feed on are also vitally important to us. These pastures can be found throughout the southeastern United States and consist of primarily non-native species such as tall fescue, bahia grass. And these species, while they're really good forage, do have the potential to become invasive in already delicate landscapes. That makes the use of native grasses alluring. However, they're less desirable because they've had a lack of selective improvement breeding. So that makes these species less tolerant of constant grazing. And there has been some research into breeding our warm season native species for forage improvements, but it hasn't been done on our cool season native grasses, such as our study species, southeastern wild rye. There's been some preliminary research into southeastern wild rye as a forage species. And here it is. This is our study species, Elymus glabriflorus. It is relatively newly distinguished from Virginia wild rye that happened in 2008. It's a native cool season grass, as I mentioned, that can be found in the eastern United States and throughout southern Canada. It's important to note that this species is mostly self-pollinated, and I'll get into that as we go on. So there has been research into southeastern wild rye for its potential as a forage species, as I mentioned. Rushing and Brian Baldwin found in 2013 that southeastern wild rye was the most adapted to forage applications when compared to other Elymus species. It does have some good quality forage characteristics such as high crude protein, low neutral detergent fiber, and low acid detergent fiber. However, it's important to note that because it hasn't been selectively bred to withstand against grazing, it doesn't tolerate grazing as much as our other commonly used species. Here it is, some illustrations and some pictures. Some distinguishing features between it and Virginia wild rye is that it has an open inflorescence. It doesn't have a boot covering the seeds, and it has long awns. I do want to point out this image to the bottom right. I'll talk about this later on when I go through my materials and methods, but this image is showing a southeastern wild rye seed, and you can see the, gosh, now I just forgot, the collar of the seed wrapped around the pseudostem. And if you can kind of see this like shiny layer right here, that's the outer epidermis layer we take for our leaf peels. So the main player of my research was mutualistic endophytic fungi. These fungi can be found in a variety of plant species, but they're most commonly found in grasses. And they have a large impact on their host plant, but it can depend on the environmental conditions as well as the host genotype. The best known and most studied species are those in the Epicloe genus, as we've heard from previous talks today. I like this picture. This is Canada wild rye with a very zoomed in image of Epicloe. And here's that same image. So Epicloe is a known systemic fungi to 80 genera of plants and hundreds of species of grasses. It grows exclusively in the above ground biomass of the plant, specifically in between epidermal cells, as seen in the image to the left. The arrow is pointing to fungal hyphae running between the epidermal cells, and it has a very characteristic convoluted, or what I like to call squiggly appearance. And it also grows in the apoplast of the seed. This other image is an isolate of Epicloe from tall fescue showing characteristic growth. So infection with Epicloe is important because it has shown to improve the hardiness of the host plant through the production of secondary metabolites. A lot of grasses lack their own biosynthetic capability to produce these secondary metabolites that can give it resistance to heat, drought, salinity, other biotic and abiotic factors. So the goal of this research was to identify and isolate endophyte from Canada wild rye, that's CWR, and then we will use our isolate to infect endophyte-free southeastern wild rye, or SEWR. And then we'll confirm endophyte infection through histological analysis and polymerase chain reaction. So before I get into my materials and methods, I'm going to just talk about really quick where we received our germplasm. I am from Mississippi State University, and we have 3 populations of southeastern wild rye that we planted. We also received 1 Canada wild rye germplasm from Shooting Star native seed to assess for endophyte infection. So the first step of the research was to figure out what germplasm is infected and what's endophyte free. I did this 2 ways. First, I did seed squashes, as seen on this slide, and I also did what's called a leaf peel, which I mentioned earlier. So to do a seed squash, we collect 10 to 20 seeds, soak them in sodium hydroxide overnight. That softens the seed coat so we can remove it. Then we place these little insides of the seeds, they're very squishy, put them onto aniline blue stain on a microscope slide and gently squash it with the cover slip. This flattens it to a very consistent layer so that we can see the apoplast of those seeds. So what you're looking at in these images are Canada wild rye and southeastern wild rye. Southeastern wild rye is this upper left-hand image, and the others are Canada wild rye. This is the apoplast layer of that seed, and the circular cells you're seeing are the apoplast cells. So I have maroon arrows in our Canada wild rye images pointing to fungal mycelia running between, it's hard to see and I apologize, running between these apoplast cells. And it's important to note that our southeastern wild rye germplasm, all of the germplasm I've looked at was endophyte free. So now that we know about our seeds that are infected or not infected, I moved on to looking at leaf peels of our already established southeastern wild rye population to get an idea of if that population was infected or not. So leaf peels are done. If you guys will go back in time to that image I showed you, we're taking that outermost epidermal layer from the inside of the leaf below the collar. It's hard to see, but here it is. We cut it with a scalpel and peel it with a pair of forceps, put it on aniline blue stain, and again, observe it under 40x magnification. The image to the left is southeastern wild rye, and the image to the right, again, is Canada wild rye. We, once again, have an arrow pointing to fungal mycelia running between those epidermal cells, and again, it's notably missing in southeastern wild rye. So we found, after peeling hundreds of leaves and squashing hundreds of seed, that all of our southeastern wild rye germplasm was endophyte free. So we do know that the plots that we used for this study had seed that had been stored previously for well over a year. There was a study that found that when a seed is stored at high heat and high humidity, the stability of the endophyte decreases over time. So our seed that had been stored in Mississippi heat and Mississippi humidity for well over a year was very likely to not have endophyte in it anymore, resulting in endophyte-free mother plants and therefore endophyte-free progeny. So now that I know that we have infected Canada wild rye, we gotta get the endophyte out. So I did this by surface disinfesting seed with bleach twice. If you're wondering about that, I can tell you about it, but I'm not gonna go into it now. Then we cut the seed in half laterally, placed it on sterile potato dextrose agar, and put it in a 25-degree dark climate control chamber. The image in the middle is about 1 month of growth, and the image to the right is about 2 months of growth. This is a very slow growing fungi. But once again, if you think back to that tall fescue colony I showed you, this is very good characteristic growth for epicloe. And I just love this image because this seed was so resilient that it's like has a shoot coming out of it despite being bleached twice and being cut in half. So I thought that's pretty cool. So we have this colony. Now what do we do with it? We're going to infect southeastern wild rye with this colony. But to be able to get the mycelia off of the plate, we had to inoculate potato dextrose broth. This was done by cutting a small section out of our colony, mashing it with a sterile pestle, mixing it with potato dextrose broth, and putting it in the dark for 7 days. This image shows the potato dextrose broth with our hyphae spheres. They're like those little speckles you see. And we did this to assist in teasing up the fungal mycelia out of the solution so we could infect the seedlings. So I hand infected 200 seedlings with a exceptionally tiny 28 gauge needle under a dissecting microscope at 15x magnification. This was a lot. It was super fun to do though. But these images are fun because you can see just how tiny these seedlings are. And this needle, if you guys know what a 28 gauge needle is, it's small and it's still much bigger than the seedling. This is one of my favorite pictures in the study. The circle is showing where that hyphae ball has been put into the coleoptile of this seedling. And now the seeds are infected. So they sit in a germ chamber set to 20-degree day, 15-degree nights for 10 days in the dark. After 7 of those days, we add a 12-hour light cycle. And then 10 days total after inoculation, the plants were moved to a 50-cell flat. Here's the 50-cell flat. Of the 200 plants that I artificially infected, 176 individuals survived the procedure, giving us a death rate of 12%, which is pretty similar to that we've found in other methodologies. So we have plants, we infected them, we got to figure out if what we did to them was successful. These are my favorite images in the entire study. These 2 images are the first southeastern wild rye plants I have seen with endophyte in them. So the image to the left is number 31. This is my favorite image. It's the golden child. I have peeled so many southeastern wild rye leaves, and this endophyte is the first time I have seen it in these plants. So great, we did some leaf peels, found out that infection was successful. So now we have to isolate DNA from these plants to be able to do polymerase chain reaction procedures. I use the CTAB method, which is a method that utilizes CTAB, which connects to the negatively charged molecules within the cell wall to make the cells burst and release the DNA. Once DNA was out and isolated, we did PCR procedures on both our artificially infected southeastern wild rye plants and the Canada wild rye germplasm that we used for infection to have a check for endophyte infection. Here's the initial PCR results. This is a gel screen I took a couple weeks ago. Sadly, I don't have more results than this because I did PCR once and haven't gotten bands again, so I'm troubleshooting that. But the maroon arrow on these is our Canada wild rye check. We use the TFA primer, which is a PCR primer that is for our endophyte housekeeping genes. So any endophyte infected sample should have bands around 860 base pairs. These pinkish arrows was a plant that I didn't know was infected because I didn't do a leaf peel on it, so that was exciting to see. Once I saw that it was infected, I did a leaf peel, and once again we have fungal mycelia running along those epidermal cells. So here's my golden child, number 31. And of the 80 plants that I've looked at thus far, we have 4 positive infected individuals. So in summary, our initial leaf peel and PCR results indicate successful infection. Latch and Christensen, whose methodology we used for this, reported a 30% successful infection rate. So we're hoping that once we look at the rest of the plants, we'll have slightly more individuals that we can work with. However, because southeastern wild rye is a self-pollinated crop, only 1 plant was needed to have success, so fist bump to that. And this is my email if you guys have any questions, but I'd love to open up the floor for questions now.
Thank you. And we do have time for a question or two.
I'm curious, like what kinds of feature you will detect with the infected plant?
We're hoping that the plants that are infected will have increased forage characteristics, so like increased tolerance to grazing and increased resistance to abiotic and biotic stressors. We're going to do a study against our endophyte-free germplasm and this germplasm once we're done with all of the PCR and leaf peel results, and we'll see what kind of impacts endophyte infection had on these plants.
You expect to transmit the endophyte through the next progeny, or you would expect to have like a seed treatment to next seeds?
Yeah, so this endophyte is transmitted vertically. So the mother plant will pass the endophyte through the seed into the seedling. So as long as we keep these seeds either in a freezer or immediately plant them so the endophyte doesn't die, the progeny should be infected. We'll do some seed squashes on the progeny just to make sure, because transmittance through horizontal transmission isn't always perfect. It's possible for a seed not to be infected even though the mother plant is, but we'll make sure that we're getting some infected seeds. Okay.
Thank you.
Thank you.
I'd like to introduce our next to last speaker. Remember, we've tacked on the presentation that wasn't able to be presented at the end of this track yesterday. Our next speaker is Sumin Wang, and I will let you all read rather than try to butcher his title and turn it over to you. Thank you.
Thanks, the chair, give me the chance to introduce our work about the sodium transport, the function in stress tolerance from a desert plant. The desert plant, Zygophyllum, distributed in the northwest China and Central Asia, with excellent adaptability to the adverse environments. Our previous research indicated that the drought inhibits the growth, but the addition of sodium chloride distributed the growth. And we also found under the drought stress, the sodium concentration in leaf increased, especially under the additional sodium chloride. And the contribution of sodium to osmotic potential is significantly increased. Our results also indicated that the ZX-NHX1 compartment like the sodium from the cytosol to the vacuole, and also significantly induced by the salt and drought treatment. And also the source one mediated the long distance transport, the sodium from root to leaves. However, how the sodium transport within the root is not clear. The many researchers indicated that the HKT transporter in the salt ecoclinic species, the most glycophytes, HKT members mainly contributed to limit the sodium transport to the shoot. But for the desert plant, the phyllum is a salt accumulated xerophyte. The function of HKT transport is not clear. Then I will introduce our results from the following 4 aspects. Our results indicated that the GFP, ZX HKT protein is targeted at the plasma membrane. And we also found use the yeast strain. This strain is the potassium uptake deficient. Compared with the typical potassium channel, AKT1, we found that the ZX-HKT1 does not participate in the potassium transport in the yeast. And we also use another yeast strain, G19. We can see same as the Arabidopsis HKT1. The ZX-HKT1 mediated the sodium uptake in the yeast. We also use the oocyte to analyze the sodium, the characteristics of HKT1. We found that this transport mediated the high affinity sodium transport. Then we analyzed the expression pattern of this gene in the desert plant. We can find either under control or salt treatment. This gene mainly expressed in the roots. And we also found that this gene was abundant within the stela, particularly in the xylem parenchyma cells. Then we analyzed the function of this gene in the Arabidopsis. We can see if we use the 33 promoters, the plant growth is inhibited. But if use the native, the Arabidopsis native transporters, promoters, the plant growth is much better. We also use the hydroponic culture we can see the over-equatorial line, the growth is increased, and also the complementary lines. We found that unlike the sodium and potassium in shoots and roots, we found in the HKT deficient lines, the shoot sodium is decreased, but in the root is increased. But in the over-equation lines is contrast the sodium. And also the trend of potassium distributed in shoot and root contrast with the sodium. Then we analyzed the equilibrium pattern of ZX, NHX in roots in the wild type zygophyllum, and also in the and found that under 50 millimole sodium chloride, the expression is decreased with the elongation of treatment time. But after under the high sodium concentration, the increased. This is matched with the HKT. With the NHX expression in leaves under the 50 milli sodium chloride consistently increased, but under the higher sodium chloride, first increased and then decreased. We also use the NHX silence lines. We can see different from this, the wild type. The expression increased with the treatment time. So from this, we can conclude that the function of the NHX essentially depends on the vacuolar sodium compactation capacity mediated by the NHX in leaves. Finally, we concluded the two. First is the NHX included a sodium selective transporter that is preferentially expressed in the root xylem parenchyma cell and pericycle cell, and play important role in inducing sodium retrieval from the xylem site. And the second is the level of the HKT is coordinated with the NHX to regulate synergistically sodium homeostasis in this plant under the saline condition. Thanks my PhD students and my colleagues. Thanks our collaborator, Tim Flau from Sussex University, the UK. Thanks for your attention.
We do have time for a few questions if we have some from the audience.
Thanks.
Thank you, sir. Thank you very much.
So that goes forward, that goes back, and this is the laser.
Okay, thank you.
We will go ahead and start our final presentation. We're glad she finally made it. Had a little rough trip yesterday, but fortunately here today. So, Wenzhuang Tang,
Yeah.
construction of alfalfa rhizobia symbiont and stability detection of their high efficiency symbiotic effects. Thank you.
Yes, thank you. It's an honor for me to stay here and make a simple presentation of my work and my team's work in the past maybe 10 years. And there is a little change in my presentation name. I'm from China, Lanzhou Gansu Agricultural University, and I'm a staff in this unit. And my cooperative tutor is Shishang Li. And today I'm going to say something about alfalfa. As we all know that alfalfa is a very important legume plant in the world because it can symbiosis with rhizobium and to transfer or absorb the atmosphere nitrogen and convert it into another form and be absorbed by the plant directly. So it's very important, but sometimes when we plant the alfalfa, we need to use some rhizobium inoculants. But due to the incompatibility between alfalfa and rhizobium and some rhizobium inoculants, so this symbiotic system doesn't always perform high nitrogen fixation effect. So we developed a solution. Maybe we can breed or develop alfalfa varieties with heritable specific high efficient nitrogen fixation effect. Yeah, it's a long system. Let me introduce. We are thinking that maybe we could breed or create a kind of seed, some kind of seed, where they containing endophytic rhizobium, which can fix nitrogen high efficiently. So we make some investigation. We have found that the endophytic rhizobium in commercial seeds differs with cultivars, places, and storage times. And we used almost 30 cultivars from introduced cultivars and native cultivars. We found that the rhizobium number in introduced cultivars are higher than those in native ones. And then we found that with the storage years increases, the number in seeds increased firstly and peaked almost as the fifth year, and then showed a downward trend. And then we also detected the number of rhizobium and other microbes in seeds. We found that with the increase of development stage, the number of rhizobium increases, and it's much more than the other microbes. So we concluded that the environment of seeds is beneficial to maintain the dominant position of rhizobium community. And then we isolated some endophyte rhizobium from seeds, alfalfa seeds, and found that they can nodulate alfalfa and promote plant growth. And then we think maybe we can utilize this kind of symbiotic function of seeds, endophytic rhizobium, and without use the rhizobium agent or rhizobium inoculant. And we are thinking to breeding alfalfa seeds containing high efficient rhizobium. So this is our goal. Maybe we can construct a kind of alfalfa rhizobium symbiont. This means construct some seeds containing endophytic rhizobium. And in these seeds are high efficiency symbiotic effects could be inherited steadily. And we use the GFP tagged endophytic tracer rhizobium. Maybe we think we could find some alternatives for the rhizobium inoculants because it's time consuming, money consuming. Maybe we can just use a can of seeds, which could maybe alfalfa rhizobium symbiont to become an alternative of inoculants. What we first do is we artificially inoculated the tracer rhizobium into alfalfa and detected the distribution dynamics of rhizobium. We found that in the root, the number of rhizobium increases with growth. And in different cultivars, we found that with the increase, with the growth stage, the number of rhizobium in seeds increases. We can see that from the vegetative stage, full blooming stage, and the pod bearing stage, the high number is emerged in the pod bearing stage. And for the part hair root contained the most number of rhizobium. And in the stem and the leaf, we find that rhizobium number are relatively stable. And most of them are found in vegetative stage and pod bearing stage. And then for the flowers, we can find that the rhizobium accumulate to the ova. And before pollination and after pollination, both in the two cultivars, we found that most of rhizobium are in the ovary, and some of them maybe in the stele, pedicle, and receptacle. And after pollination, we also can find rhizobium in all these flower organs or flower parts, except in the pedicle. This is the first one. This one is a microscopy graph we took from flower. Yeah, this is a plate. We can see that the rhizobium in the plate emerge in fluorescence. And then in the pod, we found that the number of rhizobium increased sharply in pod bearing stage, and the number are significantly higher than in the other stages, the scoring, flowering, and the maturing stages. And no matter which the cultivar is, the number is very high in pod bearing stage of the pod. And then we detected the number of rhizobium in seed. We found that with the maturity, the number of rhizobium increases significantly. And from ovule, fertilized ovule, young seeds, and mature seeds, we can see that the number increases sharply. And this is oh, sorry. Yeah. This is a picture in the seed. So we can see from this picture that we can find some endophytic rhizobium from seed, but the number is very small, not very high. So we are thinking maybe we. Oh, sorry. We then detected the number of rhizobium in dissected seed. We know that rhizobium are in seed, but which part? We find that the seeds are mostly located in seed coat and the seed extract. And for the seed coat, when we made the extract, we found that most of them are in the seed coat extract. So we concluded that rhizobium migrate and colonize seeds with growth and development. And the inner environment in seed coat is suitable for rhizobium colonization. So just as I said, the number is not very high. So we made some we think some methods to maybe we can do something to contribute to the endophytic tracer rhizobium to migrating and colonizing in seeds. We used the two rhizobium. They are all fluorescent protein tagged. And the first method is root inoculation. We choose the two methods. The one is direct root inoculation. The other one is we remove nodules from the seedling stage of the plant and then put the plant into the rhizobium solution and immersed. And after that, we replanting the plant. And 48 days later, we detect the number and we found that root inoculation benefits for rhizobium migrating from root to stem. And then at the seedling, we add some other substances. It's auxin, lanthanum chloride, and exposaccharide of azadirachtin. After add these kind of substances, and we inoculated the rhizobium into stem. The first one is smear rhizobium solution after cutting stem. The other one is inject rhizobium solution into stem pulp. And we found that after 40 hours later, the rhizobium in root and in both methods are all found in the root and lower stem. root and lower stem, and the GN5F rhizobium number is higher than the other one. And then in the flower stage, we also inoculated the rhizobium with added substances. They are boron and gibberellin. The method is flower spraying, and the other one is mulching with root watering. We found that with these methods, we can find that boron flower spraying promoted the rhizobium migrating to the stem and the leaf. And the other one is the gibberellin flowers spring. This one is higher. So these substances can contribute to the migrating of rhizobium to stem and leaf. And then in the pod bearing stage, we also use the pod spraying and root watering method to inoculate. And after that, we found that pod spraying inoculation promotes migrating and colonization of endophytic rhizobium into pods and seeds. And the best one is brown aided pod spray. You can see that we can find the endophytic rhizobium in seed. Yeah. So these seeds are containing these two rhizobium. So at the seed maturity stage, we harvest two kinds of seeds. We have a simple name. We have simple named them. The first one is seeds containing fluorescent tagged rhizobium GN5F, and they are harvested from Ganon number 5. It's a domestic cultivar in China plant after inoculated with this rhizobium. The other one is contained the 12531F rhizobium, and it's also harvested from Ganon number 5 alfalfa. So we think this is our first generation seeds. And then what can we do? We store them for 3 years. We want to find a good way to preserve them. And after 3 years later, we use the 4 kinds of storage conditions. After 3 years later, we find that for the GN5F rhizobium, the best method is envelope is the S1's condition. We can the best package material for GN5F is envelope or aluminium foil, and the best temperature is 25 degrees centigrade and 45% relative humidity. And the other one is envelope in closed bag or aluminium foil. And the best condition is 25 degrees centigrade and silicone drying. So the humidity is a little lower. And after 3 years of storation, we need to detect the stability. Maybe we want to know if there are some rhizobium left in the seeds. Maybe if they can already play their function of nodulation and nitrogen fixation. We found that for the numbers from the first seed to the plant to the second seed, we can detect the number. We can detect the 2 kinds of rhizobium in all the stage. Interestingly, we only found 1 kind of rhizobium in the second generation stage. It's 12531F. GN5F was not found in the seed, but we found them in the plants of the second generation seed. And for the detection of nodulation and nitrogen fixation stability, we found that seedling roots are preferentially infected by endophytic rhizobium and nodulate and fix nitrogen earlier. So endophytic rhizobium perform higher nodulation rate and nitrogenous activity. So we can see that the seeds containing endophytic rhizobium perform higher nodulation and nitrogen fixation ability. And for the growth promotion effect, we found that idiophytic rhizobium promote plant by almost significantly. The above ground dry weight and root dry weight are higher in our seeds containing rhizobium than the CK, which is where is no objective rhizobium founded. So we can also detect them in the plate and also in the branching stage. So we found that in all the stages, the seedlings have a growth effect of symbiotic seeds. The growth effect of symbiotic seeds exceeded that of CK. So it's a good performance. Nowadays, we have observed the third generation seed. The first generation seed was done by my colleague Miao, and the sixth generation seed was done by colleague Liu. And now we observed the third generation seed, and we also detected the number of rhizobium and detected the stability of nodulation, the stability of nitrogen fixation. They all performed good outcome. So we hope we can got the fourth, the fifth generation seed, and maybe one day, hopefully, we can breed these kind of seeds into a new cultivar and put them into production, maybe in the industry, and so on. It's hope, yeah. Our work is finished by our colleague and my cooperative tutor, Shishang Li, and thank for all of these people. And this is my email. Thank you to give me the opportunity. Thank you very much.
Thank you very much. Questions?
This is interesting work.
Yeah, thank you.
So my question is, the symbiosis can generate it, one generation, second generation, till fourth generation?
Yeah.
Do you think it is genetic modified or just a cooperation between bacteria and plant?
Nowadays, we thought that it may be, at present, it may be a cooperation result of the rhizobium and the seed. And maybe after long times of cooperated breed, it may have some genetic changes in the seed, in the alfalfa seed. But we have never identified these things. Because if it can create some genetic changes, it's a good way, it's a good thing for us to create a new cultivar. But we have never identified this.
Okay, another interesting is, can you test the bacteria in embryo, not only in coat?
In embryo, not only in?
Embryo.
Yeah.
Yeah. But you told us you find the bacteria in coat, seed coat.
Yeah.
No, do you know it is in the embryo? For example, the top meristem or root meristem.
We find that it's most of the rhizobium are located in seed coat. But we never do just like a tracer to find if it was from the ovule or some other parts of the flower. Maybe we need to do some to track the rhizobium from the flower organs to the seed.
I think that's the key for
to
test the hypothesis genetically modified.
Yeah. Thank you very much for your question.
Thank you all. Thank you.
Okay.